Hi, my name is Jamie Marshall, and I'm a senior group leader at the Broad Institute in Anna Graca's lab in the Kidney Disease Initiative. Today, I'm going to tell you about a project I'm leading to develop unbiased, near single cell spatial transcriptomics for the kidney. First, I want to introduce you to the people that contributed to this work. Taya and Xing Bo have performed the computational analyses, Hai Chi has performed the image analysis, Evan makes the SlightSeq arrays, and the technology was developed in Fei Chen's group. The single-cell data was generated by Catherine and computational analyses performed by Anna and Aishwarya in Anna Greca's lab. The human biopsies are obtained from Brigham and Women's Hospital from Anna, Astrid, Catherine, and Keith. And this work has been funded by the Broad Ignite and CZI Seed Network. So now I want to tell you why we are interested in unbiased spatial transcriptomics. Single-cell RNA sequencing has been performed on almost all tissues to gain insights between cell types in health and disease. However, these studies require tissue dissociation, resulting in the potential loss of sensitive and diseased cells, as well as information about cellular neighborhoods within the tissue. So we lose all information about tissue structure and have no way of knowing which cells interacted with each other in various structures throughout the tissue. For the kidney, this means that we have no way of knowing which podocytes, mesangial cells, and endothelial cells made up a glomerulus, or which proximal tubule cells surrounded that glomerulus. Understanding these interactions is critical for gaining deeper insights into molecular signatures of disease and heterogeneity of disease within a biopsy and between individuals. Today, I'm going to tell you about applying SlightSeq to kidney biopsies. SlightSeq is a technology that allows for the highest resolution, which is near single cell, unbiased spatial transcriptomics. SlightSeq captures the same mRNA as single-cell RNA-seq, but has the added benefit of spatial locations for every bead. In this talk, I will explain the methods that we have developed that allow mapping of kidney cell types on spatial arrays. Specifically, I will tell you about the SlightSeq workflow, improvements that we have made in SlightSeq version 2, methods for mapping cell types in mouse kidneys as well as human kidneys, analysis of mouse kidneys with tubular disease, and analysis of human kidneys with diabetic kidney disease, DKD, and ischemia, as well as conclusion and future directions. So the first step in the SlightSeq workflow is to prepare the SlightSeq arrays. An example array is shown here on the left. 10 micron beads are adhered to glass by spinning them onto liquid tape. Each bead has oligos containing a PCR handle, spatial bead barcode, UMI, oligo-DT, which binds to the polyadenylated mRNA. The arrays are then mounted into a flow cell shown here in the center. Each flow cell contains 16 different arrays. Once the arrays are mounted into the flow cell, they are subjected to sequencing, and sequencing of all 14 bases on the spatial bead barcode takes about 2.5 nights, and it's done here on a confocal microscope shown in the picture on the right. The next step in the SlightSeq workflow is to map the bead locations. The images on the top and the center of this figure shows the raw image output from sequencing of the 14 different bases, each picture is a base, of the bead barcodes. The images underneath show the computational prediction of the sequence on each bead. And these images supply the bead barcode for each bead onto the spatial arrays. Once the bead barcodes have been determined, the spatial arrays are then ready for experiments. 10 micron slices of flash frozen kidney are mounted on top of spatial arrays shown here in the picture on the top. The array is then transferred to a tube and subjected to a 15-minute hybridization in an SSC buffer and transferred for cDNA synthesis, second-strand cDNA synthesis, and tissue digestion. Once the beads containing cDNA are dissociated from the glass slide, they are then subjected to standard library preparation similar to that of single-cell RNA sequencing, including cDNA amplification and tag mutation. Final libraries are sequenced on an Illumina sequencer. Standard computational processing includes generation of a cDNA count matrix of Illumina barcodes, and then the addition of in situ bead barcodes to generate a count matrix of spatially resolved cDNA. With this count matrix, the data is ready for cell type mapping. And before I move on to that, I first want to tell you about some improvements we've made in SlightSeq version 2. As previously mentioned, SLATE-seq provides the highest resolution unbiased mRNA capture in intact tissue. SLATE-seq version 2 now provides a 10-fold increase in RNA capture compared to version 1 through the addition of second strand synthesis, better oligos on beads, and an updated in situ sequencing protocol for the bead barcodes. Now I'll show you some data comparing SLATE-seq and SLATE-seq version 2 on human kidney biopsies. Human kidney tissue often has lower RNA quality due to the time between biopsy and freezing, as well as the lack of perfusion or removal of blood. So we wanted to test whether SLATE-seq version 2 is able to generate sufficient UMIs from human tissue. Importantly, if you compare the images from the spatial arrays of human cortex on the left and medulla on the right, from SLATE-seq version 1 on top and SLATE-seq version 2 on the bottom, you will see that SLATE-seq version 2 demonstrates a hugely improved capture in the number of UMIs per bead and actually resolves some structure information. And these results suggested to us that spatial transcriptomics will be possible in human kidney tissue. So now that we have captured sufficient UMIs in human tissue, and mouse kidneys using SLATE-seq version 2, the next challenge is to confidently map cell types onto the spatial arrays. And I can tell you that this was not as simple as you might think it is. I will show you 3 methods we have currently tested in both humans and mice. The first method is NMF-reg, or non-negative matrix factorization regression. And this was the method published in the initial SLATE-seq paper. NMF-reg constructs expression of each SLATE-seq bead as a weighted combination of metagene factors of cell types defined in a single-cell RNA-seq dictionary. The second method uses Seurat clustering on gene expression from SLATE-seq beads themselves to cluster the cell types and maps them based on the bead location information onto the SLATE-seq arrays. This is identical to the method used in single-cell RNA sequencing data, but will likely suffer a loss of resolution for certain structures due to SLATE-seq beads that may contain multiple cell types. And the third method uses Seurat combined with supervised clustering from a single-cell reference. This method is similar to NMF in that a cell type dictionary of metagenes is used to map the single-cell RNA-seq clusters onto SLATE-seq V2 arrays and maps cell type projections onto the arrays. So now I will show you a comparison of these 3 methods for identifying cell types in the mouse kidney. In the NMF mapping shown here on the left, you can observe potentially 3 distinct regions in this array. One here on the top, a pink/purple one in the center, and then one here on the bottom. However, this array lacks distinct tubule-like structures and glomeruli that you would expect to see. Now, with the second method of using Seurat clustering of SLATE-seq beads themselves, you can now start to observe 4 different areas. One here, one here, one here, and one here. And each of the 4 areas contains more coherent patterns or cell structures, and you can start to imagine tubules. With the third method of using Seurat to map the single-cell RNA-seq dictionary onto SLATE-seq arrays, you can observe many distinct structures throughout the mouse kidney. In fact, this is the first method that generates an image with what we believe is distinct structures for all cell types. For instance, there's a clear distinction between the cortex, up here on the top, which is the pink and purple region, and the medulla, which is the green and yellow region. Glomeruli, which are the red, cyan, and dark purple regions, are found throughout the cortex. And vasculature, here in the dark purple, is clearly demarcated. To determine that Seurat mapping was better than NMF, we compared loadings of each cell type across the methods. The methods perform similar in regards to structure identification across the proximal tubules, shown here on the left. However, the Seurat mappings have less of these sparse background beads compared to NMF. In the distal tubules and thick ascending limb, shown here on the right, Seurat provided much better definition to the actual cell type structures, shown here in the medulla and then out here in the cortex, in addition to reduced background. And the same was true for glomeruli and vascular smooth muscle cells. Seurat provided better structure definition and reduced the amount of sparse background beads throughout the spatial arrays for both compared to NMF. Based on these results, we believe that supervised mapping of cell types with Seurat is the best method for the mouse kidney. And finally, I'm showing you cell type mappings across multiple arrays of mouse cortex on the top versus medulla on the bottom. We observe the expansion of collecting ducts, distal collecting tubules, and tau in the medulla. compared to the regions enriched for the cortex. And the cortex region had an increase in the glomeruli, which are these dark purple and red areas, compared to the regions enriched with medulla. With these results, we are confident in the Seurat mapping of cell types using a single cell reference. Now I want to show you the three methods for mapping cell types in human tissue. As I mentioned previously, the ability to map cell types in human tissue is the true test of Sleizig because the collection of human tissue is less controlled. In NMF cell mapping of human tissue, we are only able to confidently identify two structures: glomeruli in the red, the dark blue, and the light blue, and then this kind of cohesive tubular structure here in light yellow. Now with Seurat clustering of the Sleizig beads themselves, we're able to maybe distinguish four structures and more patterning of cell types across the array. And finally, with Seurat mapping using a single cell reference, we're able to clearly identify 5 structures, as well as distinct patterns within these structures on the spatial arrays. So glomeruli are in the red and the dark blue. We have proximal tubules in the pink. We have distal tubules and thick ascending limb and collecting duct in the green and the yellow. And then we have vasculature here in the cyan and the purple. And these results are greatly improved compared to the NMF cell mapping. Similar to the mouse array, we performed a direct comparison of NMF on the bottom and Seurat on the top for specific cell types: vascular smooth muscle, distal tubules, and collecting duct. The difference observed in human tissue is staggering. NMF is unable to clearly distinguish all 4 of these cell types, whereas Seurat clearly delineates cell type structures and tubules and is clearly the best method. To give you a sense of cell type mapping in human tissue, I'm showing you arrays applied to cortex on the top and medulla on the bottom from biopsies of 2 different individuals. Proximal tubules in pink and glomeruli in red and dark blue are present in the cortex, and distal tubules and collecting ducts, which are the yellow and the green, are enriched in the medulla. Clear cell type structures are observed across all arrays, giving us confidence in the cell type mapping and structure identification in human tissue. Now I want to transition to show you data collected using Sleizig to interrogate mouse kidneys with tubular disease. In this slide, I'm showing you 2 example arrays from healthy mice on the top and diseased mice on the bottom, and these arrays are enriched for medulla, as this is where the disease is present in these mice. The mouse model used has a mutation in uromodulin, or UMOD, that causes a frameshift protein which aggregates and causes disease in the thick ascending limb, or tau. In the panel on the left, there is a trend of decreased tau beads in the disease compared to wild type, specifically in the medulla. No changes are observed in the cortex. FISH validation using UMOD and CTGF, which is a well-known marker of disease processes including tissue remodeling and fibrosis, is shown on the right-hand side of the slide. Quantification of the FISH validation revealed a significant increase in injured tau cells, or those expressing both UMOD and CTGF, specifically in the medulla, shown here, which is clearly seen on the right-hand panel. In the FISH validation in the left-hand panel, you only see CTGF expressed in fibroblasts in between tubules, whereas in the UMOD knock-in mice, or the diseased mice, you see CTGF expressed within the tubule structure of the thick ascending limb. We then asked whether fibroblasts were also increased in the medulla where the injured tau cells were detected. The images on the left are from arrays of healthy on the top and diseased mice on the bottom. Fibroblasts containing beads are detected at high levels in diseased mice with tubular disease, specifically in the medulla, which is confirmed in quantification of the Sleizig beads in the middle panel shown here. So no difference in fibroblast beads is observed in the cortex, and there's an increase in fibroblast beads in the medulla of diseased mice. FISH validation was performed, and an increased number of fibroblasts were detected in the medulla of diseased mice compared to healthy mice. Finally, we looked at macrophages to determine whether they are also increased in the medulla of mice with tubular disease. In the array images on the left, you can see an increase in macrophage beads, which are the red dots, in the diseased mouse compared to healthy, specifically in the medulla. And this is quantified by plotting the percentage of macrophage beads on Slide-seq arrays in the cortex on the left and the medulla on the right. And you can observe that there is a statistically significant increase in macrophages in the medulla of diseased mice. On the far right, we validated this finding with fish using markers for macrophages. And again, we saw a statistically significant increase in macrophages in diseased mice compared to healthy mice in the medulla. In using Slide-seq to examine disease-related changes in mice with tubular disease, I have shown you that Slide-seq spatial transcriptomics detects an increase in injured tubules, fibroblasts, and macrophages, and that these changes are observed specifically in the medulla. The power of unbiased spatial transcriptomics is that we can ask questions about gene expression changes on beads for all of these cell types between healthy and diseased mice. To do this, we pooled all of the beads for each cell type from all arrays, consisting of both healthy and diseased mice, and performed clustering and differential gene expression analysis. In the center panel, I'm showing the results from this analysis where the healthy and diseased beads are mapped back onto the arrays. From the tau, which in the healthy are these dark magenta, and in the diseased beads, they are the bright pink. Fibroblasts, which are brown versus yellow for healthy and diseased, and macrophages, which are dark purple versus light purple for healthy and diseased. As you can see, the diseased tissue contains diseased beads, tau in bright pink, fibroblasts in yellow, and macrophages in light purple. And this occurs all throughout the medulla in these mice, suggesting that the entire medulla is populated with diseased thick ascending limb fibroblasts and macrophages. In contrast, in the healthy mice, the medulla is consisting of all healthy beads for these cell types. We then performed Go pathway analysis on DE genes from the diseased macrophages, which is shown on the right. In this analysis, we detected overall changes in ECM organization, Wnt signaling, lymphocyte-mediated immunity, the adaptive immune response, and VEGF signaling pathways on the macrophage beads. From this example, hopefully I have convinced you that Slide-seq spatial transcriptomics reveals disease-specific changes in a spatial manner and nominates biological pathways for further investigation. Now I would like to show you a brief example using Slide-seq spatial transcriptomics to study human kidney biopsies. On the top, I'm showing you arrays from the medulla of 3 human biopsies, one from healthy tissue, one from early diabetic kidney disease or DKD, and one from a kidney that had ischemia due to tumor compression. The large red beads on these arrays showing you all cell types are specifically showing you the location of macrophages expressing markers of activation. On the bottom, I'm showing you the percentage of beads called as macrophages on the left, and the percentage of beads expressing markers of activated macrophages, or these bright red dots, on the middle. As you can see, there's an increase in activated macrophages in the sample with ischemia, particularly in the medulla compared to the cortex. Finally, we developed a method to quantify bead neighbors for activated macrophages, or these bright red dots on the arrays. The right bottom panel is showing you the interaction frequency between the activated macrophages and other cell types across the 3 tissues. In the ischemic sample on the far right, we observed new interactions with the thick ascending limb, or tau in yellow, the fibroblasts in teal, and principal cells of the collecting duct in bright green that are not observed in the healthy sample. So in summary, I have shown you that we capture spatial arrays from both mouse and human kidneys using Slide-seq version 2. The application of Seurat cell type mapping allows us to confidently call kidney cell types from both tissues. And Slide-seq version 2 also allows for the detection of disease processes in distinct spatial locations in both human and mouse tissue. We're currently working to develop a spatial atlas of healthy human kidneys as part of the larger Human Cell Atlas efforts using Slide-seq version 2. And additionally, we're excited to apply this technology to identify molecular signatures of disease, to identify areas of disease heterogeneity within a tissue and between individuals, as well as new therapeutic targets. And finally, I just want to take a moment to thank all of the people on the slide, especially the entire Kidney SlideSeek team, as well as the rest of the Chen and Greco labs. Thank you.